Welcome to Tap Talks. I'm so glad you're all here. Looks different actually now than I had it earlier, but anyway, Tap Talks, for those who are new to us, it's part of the 500 Women Scientists mission to make science open, inclusive and accessible. We started doing these Tap Talks in collaboration with Block 15 Tap Room over a year ago. That's why it's called Tap Talks, because it goes with the tap room. And a bit of hiatus for COVID, we started doing them online, and this is going
to this. You are dead, and
thank you, whoever that was. Anyway, it was first Tuesday of the month. 500 Women Scientists. There'll be another one in April with the power of observation with Susan Salafsky, who we're just hearing. And she's got 4 hypotheses that she's going to share about evolution. So tune in for that. Just a quick little bit of housekeeping before I go forward. Just remember that check to make sure you're on mute whilst the speakers are presenting. It's much appreciated. I try and turn people on and off as we go through, but don't always. There should be a Q&A after the talk, which Ashley D'Antonio will be looking after, I hope. Is that right, Ashley? And just as a warning to everybody, this Tap Talk will be recorded. And so if you want to see it again, or you want to share it with your friends, you can check out the 500 Women Scientists Corvallis YouTube channel, which we've started doing. Anyway, it's a great pleasure. I get to introduce our trifecta of speakers tonight, with not 1, not 2, but 3, talking about coastal pollution. It's not just the microplastics. And our speakers, I'm delighted, really, it's so exciting. Suzanne Brander, she's an assistant professor in the College of Agricultural Science at OSU, the Department of Fish and Wildlife. And she's taught me so much about plastic. I mean, I can't even look at my Tupperware the same way as I used to. It's like gone. Plastic forks, knives, all gone. And so, yeah, I cry, and she's taught me so much. I'm so aware. Thank you, Suzanne. With Suzanne comes her graduate student, Sarah Hutton, also another ecotoxologist, I have to say that a few times the right way, and Estuarine Environments, which I'm probably sure I mispronounced, who is also another wonderful scientist studying ecotoxology, but also a mad big data scientist person. Anyway, please, thank you for being here. I'm so excited for this talk. I hope you have a great time. I'm going to double check that we're recording, and we are. And with that, I will hand it over and give Suzanne Brander the floor.
Great. Thank you so much, Mary, for that wonderful introduction. And thank you so much for having us to speak tonight. I'm sorry we can't be in the tap room. I miss Block 15 dearly, and hopefully we'll be back there soon as vaccinations keep moving forward, and maybe by this time next year or even earlier. So like Mary said, tonight, myself, Dr. Samreen Siddiqui, who is a postdoctoral scholar in my lab, and Sarah Hutton, who is a PhD student in the environmental molecular toxicology program at OSU, are going to be talking tonight about coastal pollution, and that it's not just the microplastics, which is probably, in terms of what you hear about marine pollution and coastal pollution these days, it's probably mostly about the microplastics, but we're going to go into a little bit more depth in that and talk about a couple of other organic pollutants that we should be concerned about. And I will apologize, Mary, I'm sorry to make you feel guilty about your Tupperware, and I'll try not to do that again. But so let's go ahead and get started. And so like Mary said, my lab group is in the Fisheries and Wildlife Department, and also part of the Coastal Oregon Marine Experiment Station here at Oregon State. And so I have lab space on campus and also at the Hatfield Marine Science Center, but I'm mostly based here in Corvallis. And so our group is focused on research in ecotoxicology, which means that we think about pollutant pollutant effects on aquatic organisms mostly. So fish, invertebrates. I have one student not speaking tonight who looks at microplastics in otter scat. So trying to get an idea of where pollutants are and also what kinds of effects, sometimes subtle effects like changes in, you know, the number of eggs a fish can lay, or changes in how swimming behavior might be happening and how that might affect an animal's ability to get its prey. And so thinking about the big picture, often how small changes in the way organisms interact with their environment can affect the bigger picture, like how many fish are out there, what does population health look like. And so broadly speaking, our ocean is affected by two main types of pollution. And so one would be, you know, what we think of as chemicals, as soluble chemicals, things that are at least partly miscible with water, not always completely. You know, oil, for example, tends to form a slick, but these are chemicals that get into the environment from industrial activities or via runoff, say pesticides due to agricultural runoff and other sources, toxic metals, maybe from mining or other industrial activities. And then of course the other big category is trash over there on the other side. And we all know that plastic is a major pollution source at the moment, and I'll talk a little bit about that in more detail. And then I'll hand the mic over to Sarah, and then she'll pass it to Samreen. And so like I said, I'll start with an introduction, talk a bit about microplastics occurrence and effects. And then Sarah is going to talk about her research on pesticide toxicity and how that can influence the behavior of fishes. Samreen will talk about perfluorinated chemicals. So if you've ever wondered what's on the surface of a nonstick pan, she's going to tell you a bit about that. And then we'll also talk a bit about solutions, and we'll leave some time for questions at the end, so we can go more into that if you're interested, or if you need more information on anything we've spoken about. So to get into the plastic problem, we all know that plastic use and disposal is increasing. You can see it, you know, you can see it every day. You can see it in the supermarket. You can see it in your own home. If you have kids, there's no way to prevent them from bringing little plastic objects home with them, you know, every day. And so we know that this is projected to continue to increase. This graph goes out to 2050, but there are similar, you know, projections don't change if you go out to 2100, for example. And as of 2015, we had about 6,300 metric tons of plastic waste that had been generated, and only 9% of that is recycled. And that really hasn't changed and isn't predicted to change, just because there aren't a lot of economic incentives to recycle plastics that are difficult to recycle because of the way they're made, what chemicals are added to them. There's all sorts of factors that complicate the recycling of plastics. A small percentage is incinerated, which isn't great either, because that produces potentially toxic chemicals in the smoke. And the rest of it is accumulated in landfills and in the environment. And this varies from country to country, but this is kind of a global average. And if current trends continue, we're looking to nearly double that as far as what's going to be in landfills by 2050. And most of the projections stop at 2050. If you think about it, 2050 really isn't that far away. And so we need to start thinking beyond that. And the US, unfortunately, we have 4% of the world's population, but we create 17% of the world's plastic waste. So that's something that we need to think about changing quickly. And, you know, as you've heard, if you've been listening to the news lately, there's probably a report out about microplastics showing up somewhere unexpected, you know, on a weekly or at least monthly basis. And so we know they're ubiquitous in the ocean, and that's really where the problem was first sort of acknowledged and discovered. But now we know they're even at relatively remote sites. And we found, there was a report out last year where they found microplastics and smaller plastic particles in the air over the Rocky Mountains. So even in isolated areas, these plastics they're in global circulation. They're circulating in the air, and so they can get to even remote locations. A lot of it is ending up on the sea floor as plastics age. Their density changes as biofilms and organisms grow on them. They tend to sink, and so there's quite a few, or quite a lot of microplastic and plastic on the seafloor. The estimate is about 14 million tons. And another recent discovery is that the ocean isn't just a sink for microplastics. It's not just where microplastics end up, but a recent paper out last year showed that it's also a source of microplastics. And so as wave action sort of stirs up that surface layer, plastics can actually be ejected back into the atmosphere from the ocean. And so really just, I bring this up just to convey that these plastics are, everywhere we look, and they're not staying in one place. They can be distributed quite easily, particularly the smaller size fractions. Right. And we also know that they're transferred through food webs. But one important thing to mention is that, we know that persistent chemicals, think about something that was used years ago but is still around, like PCBs that were used in electronics, or DDT, made famous by Rachel Carson's book, Silent Spring. We still find those chemicals because they're really persistent, and they hang around in fat tissues. We find them in animals and in humans, but plastics are a little bit different in that they don't bioaccumulate in the same way. And so generally scientists are finding more plastics in the smaller organisms, in the zooplankton, in the little copepods and small shrimp and other prey items that larger fishes tend to have in their diet. They don't necessarily, you don't necessarily see this linear trend of bioaccumulation. And so you're not necessarily going to find more plastics in the larger organisms, but all that being said, we do know that we do get some exposure via our seafood. And this is what this graph is trying to convey, just showing that you have direct accumulation from the water across organisms, but they can also accumulate it from their prey. And we do know that that there is not a substantial, but a considerable amount of plastic in our seafood. Maybe it's not as much as we're getting exposed to in, say, other sources like drinking water or air, but it is a route of exposure, especially for microfibers. There was a study out a couple of weeks ago showing that microfibers produced likely from the washing of synthetic clothing in the US and other countries, such as Canada, are actually getting into ocean circulation and atmospheric circulation and ending up in places like the Arctic Circle, for example. So just it's definitely ubiquitous and is being found everywhere we look. And a comment I often get when I talk about plastic pollution is, well, plastics, that's really a challenging problem, but climate change isn't that much worse, and shouldn't we be focusing all of our resources on that? And I agree that climate change is a more challenging problem, and we should be focusing our resources on that. But all that being said, you can kind of think of plastics and climate change as being part of this larger addiction that we have to fossil fuels. And so this graphic I really like because it kind of puts it in perspective. If you go back to 2014, plastic's share of global oil consumption was about 6%. Predicting out to 2050, if we continue this business as usual pattern, it's going to be more like 20%. And so really, plastics are just part of this larger issue we have with this addiction and reliance on an unsustainable resource. Also made from fossil fuels. Plastics are mostly made from natural gas. And although there are a lot of papers out on microplastic effects, we're still in the process of trying to figure out what the ultimate effect is on aquatic life and also on terrestrial life, and just responses in general, responses in humans. That's an area that's still really, really in need of a lot of investigation. But what we probably know the most about are effects on aquatic organisms, so fish, crustaceans, other mostly marine animals and freshwater animals as well. And what we're finding is that for microplastics, and these are plastics that are less than 5 millimeters in one dimension, and often much, much smaller than that. So the research that my group does often looks at plastics that are, smaller than 100 microns in size, because those are the size of debris items that smaller organisms, larval fish, or other aquatic organisms like zooplankton are going to interact with. But what we're finding as a broader research community is that the size and shape of these plastics really matters. The smaller plastics can sometimes be more toxic because it's easier for an organism to ingest them, or for them to become entrapped in tissues, in the gut, or to become stuck in the gills, for example. And shape matters too. Studies are showing that fibers can be more toxic than other plastic shapes, like spheres. If you think of a microbead that might be in your face wash, although that's been banned, so probably not anymore, but think about how that used to be. Or a fragment, say, that's coming off of a bottle that's broken down in the sun. Fibers look to be more toxic, and the shape of the plastic can also affect whether it's accumulated or not, or whether it can be excreted by an animal, how long it stays inside of an animal. And generally, what we're finding with plastic exposure is that it causes kind of a general oxidative stress response. What's oxidative stress? Well, oxidative stress in your cells is going to produce reactive oxygen species, and reactive oxygen species can cause structural damage, and they can also eventually lead to more severe effects. And so other effects of plastic exposure, for example, are things like effects on reproduction or growth. If an animal ingests a plastic item and it gives a sense of kind of false fullness, so they don't ingest other, prey items or other food that would be of nutritive value. So, and another question I often get is about the pollutants that stick to plastics or absorb to plastics. And the jury is kind of still out on that question, because we know, without a doubt, that a lot of the plasticizers and other additives used in plastics can be harmful and can be toxic at high concentrations. It's difficult to tell whether a small amount of plasticizer on a tiny piece of plastic that's ingested by a fish is really going to be enough to cause a toxic effect on its own. And it's really a little bit more complicated, because the animal is being exposed to the plastic and the pollutant at the same time. And sometimes it takes a long time, or it takes a high amount of pollutant to actually cause an additional effect. And so in a lot of ways, it seems like just the presence of this three-dimensional object, this foreign object, is more of a concern than the pollutants that might be coming along with the plastic. But there's still a lot of research in that area too. So I'll try to finish up here so I don't take up too much time from the others, but just also want to hit home the point that when we're talking about microplastics, we're talking about truly tiny plastics too. You can think about the 5 millimeter and less, but this is an animal that I did some work with a couple of years ago when I was faculty in North Carolina. And it's a tintinnid ciliate, single-celled. It's about 100 to 120 microns in size. And it's at the base of freshwater and marine food webs. And so larval fish and other small organisms eat it. And what we found in the lab was that it will readily ingest, or I shouldn't say ingest, it takes in these particles into its cellular compartment. And it's not able to egest them. And so the fish were accumulating more plastics from these tiny ciliates than they were directly from the water. And so just it hits home the complexity of this, that it's affecting animals at the base of food webs, and then those plastics that maybe are too small for a fish to see are going to enter the food web via their prey, even their single-celled prey. And then lastly, just to talk a little bit about some research that we're doing here at OSU currently. A big focus of a lot of research groups in the microplastics field right now is on tire wear particles. You may have heard about the study that was completed up in Washington about tire wear particle leachate being toxic to salmon. That came out a couple of months ago. And here we're looking at the actual physical wear particles themselves and creating those in the lab, and then exposing larval fish at different concentrations to see if it affects the way they interact with their environment and the way they behave, which can scale up to effects in the environment because they may not be able to avoid prey or to avoid predation or to obtain food. And so these are fish. You can see these tiny little larval fish swimming around in these wells. And this is a behavioral assessment system that Sarah is going to talk a little bit more about. She's also using it for her pesticide studies. But we're able to do these pretty high throughput studies where we can look at lots of different concentrations of plastics at different salinities to see how that affects the fish. And then that gives us the types of data that are needed to actually assess the risk of what microplastics might be doing to fish in the environment. And so I'm going to finish up there and happy to address questions. I know that was a lot of information, but I'm going to pass the baton to Sarah, and she's going to talk about pesticide toxicity. So I'll let her go.
Thanks, Dr. Brander. All right, so we just heard about microplastics. So super small and tiny, pretty hard to see with the naked eye, but you can see them if you get them under a microscope. So now I'm going to kind of transition and talk about pesticide toxicity in estuaries. And this is something pretty invisible to us, so we don't see it when we're just looking out in the environment. So you wouldn't be able to see that type of contamination just by making a visual observation. So a lot of my work focuses on the estuarine environment. As we saw from the slide, the estuarine environment, and Dr. Brander, I don't know if, thanks. So the estuarine environment, as we see from the slide, is really where the river meets the sea. So this is definitely a coastal pollution problem that I'm focusing on, where fresh water flowing in from the rivers meets salt water from the ocean. And if we go to the next slide, we can see that there are gradients of salt concentration within estuarine systems. And so the ocean, if you've swam in the ocean, you kind of get that in your mouth. It doesn't taste very good, although the river probably doesn't taste very good either, but it's really salty. And we measure this in parts per thousand, which is the concentration of salt. So in the ocean, we're looking at about 30 to 35 parts per thousand, depending on where you are. And in an estuary, we can be looking at anywhere from 0 to 30 parts per thousand. And what happens as we move further from the river, oh, if we could just go back to the last slide really fast, thanks. What happens as we move further from the river is it gets saltier, but we see these different zones being formed with the water as the density and the salt concentration changes. So as we have more salt, the water actually becomes denser. And so we have these stratifications or these zones within estuaries. And different organisms can live within these different areas in the estuary. And so this is important for me to consider in my own research, because I really focus here at the 0 to about 15 parts per thousand. So kind of in the more fresh area of the estuary. And this is also a very sensitive area because a lot of organisms move into these lower salinity regions to spawn or have offspring. And so we can have larvae and smaller baby organisms living there. And then as they get older, maybe they move into those higher salinity areas. So next slide, we can talk about how this is important for thinking about pesticide toxicity. So when we just have fresh water, for example, just pure H2O, right? The water molecules are able to move around really easily around each other, and those pesticides can dissolve more easily into that water. But as we have salt added and the ionic concentration increases, the water actually becomes more rigid. And so those chemicals aren't able to dissolve as much in the water, and they're pushed from the water column and can enter the organism. And so as we move from that freshwater area of the estuary to those higher salinity regions, we can have changes in the way that these chemicals are interacting with the water and with the organisms. Next slide. So how do these pesticides even get into these estuarine environments, right? Well, a lot of this can happen at home with our own personal care products, such as sunscreens or maybe your makeup. Pharmaceuticals that you use can also enter estuaries through sewer systems and things like that. But what I really focus on are pesticides. And a lot of this is due to agricultural use, although some of this is also from our own personal uses at home. So things that you spray on your lawn, for example, can run off from your lawn or from farms when it rains and can get into rivers and streams. And then, as we learned from Finding Nemo, all drains lead to the ocean, right? So you were here for trivia. We learned that marlin maybe should have been called Marla, but this quote actually is true for the most part. So there can be events happening, pesticides and things that are used further away from the ocean that actually end up reaching these coastal regions just through transportation within the environment. Next slide. So as I mentioned, my research focuses a lot on pesticides, and one specific class of pesticides that I work with are pyrethroids. So pyrethroids are a synthetic pesticide. They're used mostly as an insecticide to kill insects. So they can sometimes be found in things in the pet store to treat fleas, or maybe you use it at home to spray for aphids on plants that you have. But these are synthetic, but they are derived from something called pyrethrin, which is also available. This is just a picture that I took from Amazon, so you could order that online. And this photo of the pyrethroid, which is the synthetic version, is taken from eBay. So these are definitely available for purchase, widely on the internet. But the pyrethrin, which is the more natural version, is derived from chrysanthemum flowers. So here we see chrysanthemum flower. And these are relatively non-toxic to humans. However, they're about 1,000 times more toxic to insects and are also very toxic to fish as well, as I'll talk about in later slides as well. So as Dr. Brander mentioned at the beginning, I focus a lot on behavior and using behavior as an endpoint for studying pesticide toxicity. And there are a few reasons why behavior is a really good endpoint to look at. The main one that I think about a lot is how sensitive behavior is. So as we'll see from some of my results, I look at very low concentrations of pesticides because we want to be thinking about what is actually out in the environment. And even at these low concentrations, we see changes in behavior. And so we can have a high confidence that we are detecting toxicity. So if there's toxicity to see, we're going to find it. The other reason is that it's extremely ecologically relevant. So sometimes, especially at these earlier life stages, organisms exposed to pesticides and other toxins can have developmental deformities or neurological deformities as well that can influence behavior. So we can see bent spines or tails and things like that that can affect their swimming. And when we see changes in behavior, as was kind of mentioned earlier, this can affect a fish's ability to feed or avoid becoming prey, or also can affect reproduction. And so all of these changes can change how a population is able to withstand pesticide exposure. As you can imagine, if you're not able to reproduce, you're not able to have offspring, and so a population could decline in the wild. So thinking about this in the lab, so we think a lot about big picture, what's happening out in the environment, but then we bring it back down to a smaller level in the lab so that we can study this, not out in the environment, right? So I really think about environmentally relevant concentrations of these pesticides so that it's applicable to the real world. And so a lot of these that I study have been found out in the environment at 1 or 10 nanograms per liter. And just to kind of put that into perspective, that's 1 part per trillion, which is equivalent to a single drop of water in 20 Olympic-sized swimming pools. So it's very diluted, and it's kind of hard to even like imagine how diluted that becomes out in the environment. And so that's really the concentration that my work is focusing on, these very low concentrations. And then in here, we just see a picture of the fish that we use to study them. So I think they're kind of cute when they're that small. There they are. So again, thinking about the lab. So this is the machine that we use to study behavior. So not quite as easy as just sitting there and watching them swim around, although I do a lot of that as well. So we have this thing called a Noldus ethovision, and there's a light that turns on here at the bottom. Dr. B, if you just I think I had an animation on this one as well. Yeah. So there's a light that turns on here, and then the fish go in a tray down at the bottom there. And so we can track them using a camera up here at the top that shows, that captures video of the fish swimming around in the bottom tray. And then the tracking software attached to this computer is actually able to turn all of that video into numbers that I can then use to analyze to really understand on a quantitative level, like what is happening with the behavior of these fish. Next slide. Okay, so just getting into some results of what happens when we expose fish for only 96 hours. So these are pretty short exposure times to these really low concentrations, like we talked about, at these salinities that are closer to that freshwater side of the estuary. So 0.5, 2, and 6 parts per thousand. And there's a quick animation on here as well. But what we see is that there is statistically significant differences in the toxicity of these pyrethroids at these different salinities. And so I'll just kind of walk you through this so that we can see how salinity affects the toxicity of these compounds. So what we're looking at here are the number of seconds that the fish are frozen for. So you can imagine that if a fish is out in the environment and it's not moving, it's more likely to be eaten perhaps, right? So at this 1 nanogram per liter, so again, 1 drop in 20 Olympic swimming pools, the fish are frozen for longer, so they're not moving relative to the control, which we see here in red. And then as we move up in salinity, these even higher concentrations start to become a problem. What's really interesting here is that these salinities at 0.5 and 2 parts per thousand, the fish freeze more, but as we get up to the 6 parts per thousand, they freeze less, so they become more active. And we see differences for different pyrethroids, so it's not the same for every pesticide. It can change depending on the pesticide. And so for this one, they're frozen less at this 1 nanogram per liter concentration at the 0.5 parts per thousand. But then in the 2 parts per thousand, they're frozen less throughout all of the concentrations. And so what's really cool about behavior is that we're able to detect these subtle differences in concentration and salinity just by looking at behavior alone. So if we move to the next slide, just kind of summarizing like why behavior is such an important thing to be looking at in terms of ecotoxicology. So again, it's really relevant to what's happening out in the environment. And pesticides can have an effect on different at different salinities. And so like we kind of talked about, this can have an effect on organisms that maybe spawn at these lower salinities and have offspring and larval fish there. And that can have real implications because those are really sensitive life stages. And so something that's happening to a larval fish can have large implications for that fish as it gets older and moves into reproductive stages of its life. And again, this is just really relevant for types of fish that are endangered and things like that. And so that's why we think a lot about this, these different salinities. And with that, I'll pass it over to Samreen.
Okay, hello everyone. So I'm taking over for PFAS compounds, that's perfluorinated compounds from Sarah. These are the compounds also called forever chemicals. Just because of their nature, they never go away from the environment. They are man-made chemicals and introduced in US in 1940s. They are highly persistent in environment as well as in human body. That means they don't break down. They just accumulate over time. And as an estimate, 6,000 chemical compounds actually belong to this PFAS family. On your right, this is the structure, chemical structure showing like where each carbon and oxygen is, just to show you what a PFAS compound look like in chemistry. And below the figure is showing PFAS sources. So mostly you find them in non-stick cookware, the black coated cookwares. Stain resistant product, like anything which can resist the stain. Firefighting foams, they are the biggest one, and they are more in the news nowadays because of the cancer issue causing in firefighters. Next is photography, in shampoo, in pesticides, in paints, fast food. Like they are, if you think, they are everywhere. Like they are in low quantities, but they are everywhere. That's why they call forever chemicals and are more taking more attention nowadays. So if you move to the next slide, I will take you through how they evolve. Just to give you a brief history. So they actually discovered accidentally in 1938. Then they moved to, the company was Teflon, and Teflon used it in their project in 1940s. Then they used in 1950s in the non-stick pans, and they find really good results, and they didn't discover the toxicity that time. Because of their nature of extremely persistent and very good chemical qualities, they get more attention and get involved in aqueous film forming foams, also called AFFs, in 1960s, and from there they developed into the more firefighting things. In 1970s, they started including it to everywhere. So their industry expands to all other products which we have nowadays. In 2000s, it distributed globally and just went out of the reach. And then at current, they actually increased public scrutiny because of their nature of never dying and never removing. They are like taking lawsuit settlements and their development, and they are trying to get new chemicals other than this. PFAS production actually banned now. They are not producing anymore. They are changing or switching it to other compounds, which can be more toxic and which are more toxic, which from their previous PFAS, and they are going under like what can be the best, but still they are there. And if I will move to the next slide, I'll show you like from where they come from. Now, you know, they are mostly everywhere. So major sources of there to the surface water, which then ends to the ocean, is first of all from the landfill. Everything which goes on the waste ends in the landfill. It leaches out to the water with rainfall, with air, with anything you can think of in the nature going on. It just leaches out in the surface water. Firefighting foams from military bases, airports, and other places are the major source. Other than that, and third major source is their manufacturing industries who are manufacturing it. Finally, they are ending up into the surface water, including wastewater. So whatever we use in our home, that goes into the wastewater and ends up in the surface water and goes into the coastal water or marine systems. And if we move to the next animation, I am going to talk a little bit about like, that's what I explained in earlier slide. They are no longer manufactured in US. And 8 companies, they did major commitment. There is no legal foundation for them now to produce, but they just committed, like 8 major companies, they just committed that they are going to end up PFOAs production by 2015. And US in particular don't have any critical guidelines yet, although this was a big issue in North Carolina recently, and that's from there it got most attention. However, right now EPA got changed its leadership and they are working on it, which I will speak more in the later part. If you can change the slide. I'm going to explain a little bit the work I have done based on this information on my research from here. So my whole objective of my research was these PFAS compounds, they are actually coming from the wastewater treatment plant, ending up into the surface water, running through the ocean or coastal system, you may say, and there are aquaculture facilities actually taking water from the surface water, and other aquaculture facilities actually have their fishes onto the coastal system. So they are actually into the water that contains PFAS. That PFAS unintentionally taken up by the fishes, and when we eat those fishes, they just get to us. So that was the whole objective or aim of my research, which I did, and I'm going to share my results with you, like what I did. I took the Genex as a chemical, that's another PFAS compound, which came in replacement of PFOA compounds. And Genex, it was like get most concern attention actually in June 2017, when it was found in Cape Fear River in North Carolina. And that's where it got mostly like what it is, from where it is coming from, since it is in drinking water system of these. And just to introduce you, PFCs, they are having much health concern. They can cause thyroid diseases, lower birth weight, liver and kidney damages, and many others problem. And I will explain more in the later slides. In my research, what I did is I did 96 hours of exposure of the Genex, followed by 96 hours of their clean water. So I want to see in this research, once we expose it to Genex, what are the options for the farmers to make it clean? So is there a way farmers can clean the fish after they get exposed to Genex? For that reason, we kept them in clean water for 96 hours. We collected their liver, plasma, carcass, and fillet to analyze what is their bioconcentration is. Bioconcentration is the amount of Genex they are accumulating in their tissue after taking it up, how much they took the Genex, how much they released the Genex, and what was their biological life, like how long the Genex was present in the fish when we leave them into, before we leave them into the clean water. And we did it at, Sarah explained you like about the salinity. The marine salinity or seawater salinity is 35 ppt, and we kept them at 0 and 16 ppt. Since tilapia is a fish which can go from 0, from fresh water to extremely high salinity up to 60 ppt. So our main aim was to see like which one is more toxic, is saltwater toxic or is freshwater toxic, and what it is doing in there. So to the next slide. What we found, this is the bioconcentration results, and blue is liver, green is plasma, red is fillet, and orange is carcass. Carcass is whatever left off of the fillet and plasma and liver we collected. Before the dotted line, it's uptake, how much they took Genex when we kept them in Genex for 96 hours. After the dotted line, it's depuration, when we kept them into the fresh water, what they did. So if you see just in a general point of view, the bars are actually increasing in trend. And then when we keep them in depuration, the Genex is releasing out. So it's good, like farmers can do something to save themselves. And if you see the order, they are mostly highly concentrated in plasma, that's in the blood of the fish, followed by liver, then followed by carcass and the fillet. And the good news is when we eat fish, we mostly eat fillet, we never eat plasma. So it's a good part that we are leaving that part out. Liver is there to do its job. So liver is our organ in the body which takes out the toxic chemicals. So it's doing its job. That's a good point. That's why it's bioaccumulating more. And carcass is the one which is like on the margin, and it can take like, it can have anything in it, and we hardly anyone uses carcass. Fillet was the one which was having least one. However, if you see the trend, it is stays the same. It's not decreasing like liver and plasma actually drop down. Fillet is not doing that, which I will explain you more in next slide. So when we compared between the 0 salinity and 16 ppd salinity, what it's doing, we didn't found much differences in carcass except a few in the beginning when it was uptaking. When we moved to the fillet, we found really big differences between the 16 ppt, that's the highest one, compared to the fillet at freshwater. So when tilapia is coming from the freshwater, it's not bioaccumulating that much of Genex as compared to the saltwater, which was a really interesting thing for us. When we moved to the plasma, we found like similar differences. Saltwater is actually bioaccumulating more compared to the freshwater. But we are not worried that much about plasma. And in the case of liver, we follow generally the same trend, but not all around. It's mostly overlapping. So liver is doing equal job in both the parts. To see what is happening, how long they are bioaccumulating, we see in the red fillet actually taking longest to depurate. So even when we left the fillet in fresh water for more than 96 hours, so this is like 54 days according to our calculation. Fillet took 54 days after exposing to Genex to release out most of its Genex in it, which is a long time. None of the farmer has that many resources. They can keep 54 days of fish into the fresh water, and keeping filtration and everything, it may bring cost very high. Followed by carcass, plasma, and liver. Liver is the one which is depurating very fast, but no one is eating fish liver that much. So that's one of the biggest concern we found with the Genex. If we'll move to the next slide. The next concern we found is at 96 hours, we did a quick calculation. Fish fillet concentration was 0.312 microgram per liter. A person usually eat 100 gram of fillet serving at a time. And if you multiply that by 100, the concentration we found, it's 31.2 microgram of Genex, which is so enough to affect a single person. Even we found them in such a low quantities, they are enough to affect a person. And if someone is eating more than one fillet, they are going to actually bioaccumulate Genex more into their body. So that's another concern we found with this chemical. So why should we care? First of all, if it is retaining PFAS longer in saline water, so that's a point of concern. Second point of concern is Genex has really high life in the muscles tissue. That's 54 days, that's long to depurate. And farmers don't have that much resources to culture fish in that clean water and contaminated like wild caught fishes. How you are going to catch fish in the wildlife and going to keep it in the clean water, that's not possible for most of the farmers. So what we can do to save ourselves and what is going on and why we are concerned. So even if we expose to PFAS, these are highly risk in adult, in children, and in pregnant women. In adults, it can cause risk of thyroid disease, increase blood cholesterol level, decrease the body's response to vaccines. In pregnant women, it can cause decreased fertility, increased risk of high blood pressure and preeclampsia, and in the lower, in children, it can cause lower infant birth weight. So it's a concern, and it was from the North Carolina government organization. That's what they are doing, and EPA is working on it. Being an individual, what we can do to keep us safeguard is like a couple of ways. We can use filtered water, but not every filtered water works the same. I am going to talk like briefly about three major ways. First of all is granular activated carbon filter. That's normal charcoal filter. It's an excellent removal of PFAS, but it doesn't remove all the PFAS. So there are two kinds of PFAS, long chain and short chain. As the name suggests, it has more carbon, and another one has less carbon. So that's a disadvantage. It won't remove fully, but it removes partially. Another way is ion exchange. That's there are not all ions product actually that's with everyone, every filtration system. Not all actually remove the PFAS, but most effective, they can remove PFAS. And third one is membrane technology. That's more commonly called as reverse osmosis or RO water or of nanofiltration technique. And it is actually expensive, and it may increase. It's good for industrial or big places, but being in home, it's a little cost effective, energy demanding. So out of all those three, charcoal filter is the one we can actually introduce in our homes and keep ourselves healthy. And with that, I think I'm almost here, and I will pass on to Dr. Brander again to conclude the whole presentation.
Here I am. I was going to let Sarah cover this slide, but I'm also happy to chat about it as well, since this was her slide that she put together. But just, it's always good to talk about the solutions, and I'll go ahead and let Sarah cover this one, and then I'll get to the others.
Okay, sounds good. Thanks, Dr. B. So there's kind of this rhyme that you hear sometimes in chemistry or toxicology. The solution to pollution is dilution. And when I was in undergrad, they told us that in our chem labs, you know, like, oh, it's okay, just dump it down the drain. It doesn't matter. It's going to go into the wastewater treatment plant and be fine. But I think one of the big takeaways from my work is that that's not necessarily the case. As we talked about, these are very minute concentrations of I don't want to be alarmist in any way, but I don't think we should be telling, like, it's not always that dilution is the best cause or the best solution, right? So mitigating our use of pesticides is just a really good habit to get into. And, you know, I'm happy to talk about all of the ways in which pesticides can be useful as well. So by no means am I advocating to stop using them completely, but there are a number of things we can do in our daily lives to kind of shift these habits that we have. And so I think just being mindful of greener ways that we can be at home. So instead of maybe getting that ant spray, you know, you can use diatomaceous earth, or instead of spraying a pesticide, you can get ladybugs shipped to you in the mail, and you can put them on plants at home. And the other thing that I think is important to take away is that there are organizations that are working to understand effects of pollution in estuaries and on fish species in many different bodies of water. And so people out there care, and if you care to, you know, use your voice and vote and just be involved in those sorts of advocacy programs.
Absolutely. And just another thought to add on there is if you can, and it can be more expensive, buying organic, at least for crops that tend to use the heaviest amounts of pesticides, is good, not just for you, but also for the farm workers applying those pesticides who get exposed to really, really high levels of those chemicals sometimes. Back to plastics and thinking about solutions on that front. And of course, just like pesticides, we're not going to be able to completely stop using plastics. They have lots of life-saving uses, for example, and we need to figure out a way to coexist with them and rely on them less than we are right now. And California has really been moving forward in that direction, and has a couple of pieces of legislation that were passed in 2018 that are driving forward the process for regulating microplastics in drinking water. And then a few years from now, they're also going to be looking at assessing the risk of microplastics to marine and estuarine ecosystems in that state. And it's likely that the direction that California is moving in will influence movement at the federal level and maybe in other states too. But they've recently put together the world's first regulatory definition for microplastics. And by this summer, they are going to begin regulating microplastics in drinking water. So they'll be required to monitor how many plastics are in drinking water coming from different municipalities within the state. And then another bill that is currently on the table that hasn't been voted on yet is one that would require all washing machines produced, see, it would on or before January 1st, 2024. So it would have to be by January of 2024. All washing machines sold as new in the state of California would be required to contain a microfiber filtration system. And this has been shown in some recent studies to really reduce the amount of microfiber contamination, a lot of which is synthetic. So polyester, rayon, all of those convenient materials that don't wrinkle that our clothes are often made of, are producing a lot of contamination. And this is just a filter that simply is installed on your machine that catches those fibers and you dump them in the trash, rather than having them go to a wastewater treatment plant and then either getting discharged in the effluent after it's treated or sinking out into the biosolids, which are then often amended and sterilized and then reapplied to agricultural lands as fertilizer. And so it's another way that fibers get back into the environment. So a really kind of simple and pretty efficient solution that would really reduce the amount of microfibers loading into the environment in California. And then maybe eventually it's something that's even been discussed in potential federal legislation as well that might be coming out this year. So with that, we all three of us are happy to take questions on anything we talked about today. I realize we went on for a little bit long and want to post the slides so you have our websites where you can get more information. If you're interested in learning more, please follow us on Twitter. We also have Facebook and Instagram accounts for our microplastics research. Pictures of some of our wonderful colleagues, but definitely there are many, many more that we could acknowledge. It's definitely, this kind of work definitely takes a village and lots of great collaborators. So happy to take questions and thank you for listening.
Thank you.
Thank you all. Oh, go ahead, Mary. I was gonna.
All right. Before Ashley jumps in, I just want to say thank you. Just to point out to people, there's a reaction button at the bottom of your Zoom. Hit it. like give lots of thumbs up, hearts, all the things that you need to do. Now's the time to do it. Put them by your name so that we know you're there. Big thank you as well to Suzanne, Sarah, and Samreen for a fabulous talk. And I always learn so much from that. Before we get into Q&A, just a quick reminder, next month's TAP Talk is actually April 6th, I think. First Tuesday of April, going to be from Susan Solafsky, and she's going to really take us through the process of how to use observation and ecological principles to identify mechanisms of life on Earth. For me, this actually means that I'll never be able to look at a chicken again without thinking of T-Rex. So join in on that because it will probably change your perspective on life, the world, and the universe, and it will be brilliant. I also, before I let Ashley take over, also want to give a big shout out, well, to Ashley because she's doing Q&A and she's phenomenal, but also to Yvonne Reicher, who's done all the social media for TAP Talks. So she's been the one sending out all the emails and social media, Instagram, Twitter. So if you see Yvonne, give her a virtual hug or an air hug or whatever you need to do. But there's a great group of people at 500 Women Scientists who make this happen. And I'm indebted to them. They're wonderful, fabulous people, including Suzanne, who used to be a pod leader for 500 Women Scientists. And so give her a hug as well. Oh. Before I let Ashley get it, because I'm here and I'm talking, I'd just like to ask a question. I think it's probably to Samreen. I should put my video on, which is like, when we, so I love the microbiome. People who know me know that I'm obsessed about the microbiome. But when we ingest as humans all these little microplastics and stuff that go in, do we poop them all out or do we accumulate them? And where do we accumulate them?
No, I can speak to that a little bit. It's something that an area I've become more interested in recently, thinking about the evidence that's coming out as far as where microplastics are showing up in human tissues. And it turns out that we do, of course, excrete quite a few of them, but some of them are getting caught in the lining of our intestine or in the lining of our stomach. Some are able to translocate if they're small enough. There have been studies in mice that have shown that very small microplastics and nanoplastics can end up in the liver, for example, or in the reproductive tissues. And so they're definitely a part of us. They've been found in the placenta too. We just don't know exactly what the long-term effects are yet, but there is evidence from mouse models that they do cause inflammation in the gut, and they can lead to alterations in the microbiome with longer-term exposure. But still, lots of research to be done in that area. So yeah, it's a really good question. And I knew you were going to ask a question about the microbiome, but I'm glad you did. But Samreen or Sarah, did you have anything to add to that? Okay. Good.
All right. Well, if folks have questions, you can put them into the chat box and then I'll facilitate. And I'll just pose these questions to all three of you, and whoever wants to take it can go ahead unless someone has a specific question for one of you. So the first question is from Brian, and Brian says, do the microplastics, especially the ones floating near the air-water interface, absorb the pesticide residues that are in the water?
That's a really good question. And the answer is that we know for some pesticides, like DDT, for example, which is very what we would call lipophilic or hydrophobic. So it doesn't mix well with water. It wants to hang out with plastic because they're more similar to one another. And so DDT is found to absorb onto plastic. So the answer to that is yes, they do. But the question is it enough to actually cause an effect in an organism? So if a fish ingests a piece of plastic that contains pesticide, is the plastic going to hang out in its gut enough that that pesticide leaches off and causes some sort of effect in the animal? Or is it just that it has this three-dimensional object that is not biodegradable in its stomach, and that's what's causing it to eat less and maybe to then grow less or to be less likely to reproduce? So yes, but it's complicated.
Great, thanks. Also, I was wondering if you could unshare your screen so we can see your faces.
Oh, sure.
That would be wonderful. Oh no, it's okay. Don't worry about it. I tried to see if I could do it for you, but I couldn't.
Okay, I just unshared.
Thank you. So we have a question from Natalie, and Natalie starts by saying this feels like a possibly ridiculous question, but thinking about the microfiber situation, does washing and reusing plastic bags, Natalie's saying she literally has decade-old Ziplocs in my collection, contribute microplastics to water too?
I mean, they probably do, but it's probably better to reuse. The overall amount of plastic you're producing is going to be less if you reuse, and maybe those bags shed a little bit, than it is if you're purchasing new bags. And I will say, we in my house, we must have like 20 bags that are in circulation that we wash and reuse. So I can relate to that for sure.
Thanks. Nellie says that makes her feel a bit better.
You're doing a good thing.
While we wait to see if anyone else puts in a question, I have a question for all of you that I like to ask folks. What inspired you to start doing this research? What got you interested in this line of research?
Yeah, I'll let Sarah or Simreen take that one.
Yeah, I can go first. I think, I don't know, it's kind of just like something that I I don't want to say that I fell into it. I grew up in a pretty agriculture heavy town, so I was surrounded by pesticide use growing up and I saw a lot of it. And then I went to college and I didn't really know what I wanted to do until I took a course on marine pollution and developmental toxicology related to ocean pollution. And I don't know, it just kind of all clicked and I fell in love with it. So here I am.
So I'm from India basically, and I have seen lots and lots of pollution going on around, but I never thought much about it until I went to UK and I saw this is the clean world. We can see clean worlds. I really wanted to do something to help our place to be better. And then I just get into this and I'm still learning. And I think I will be in a position soon to do something there to reduce the pollution.
Yeah, and I can say along kind of similar lines to Sarah, I didn't necessarily know, as a kid that this was the line of work I wanted to get into, but I always had a concern about pollution and environment and was interested in environmental protection. And I started in college as a political science major because I was thinking of being an environmental lawyer. And then I took my first law class and thought, no, this isn't for me. And so ended up drifting over to science, and here I am now.
Great, thank you for sharing those stories. We have a question from Greg. Greg asks, are particular types of plastics worse for ecosystems than others?
Yes and no. I mean, we're still trying to figure that out. And I've actually been part of this months-long process through the state of California where a nonprofit research group there has compiled a database of all of the toxicity studies that have been done on microplastics so far. And then they've put together this app that allows you to compare toxicity based on shape and size and plastic type. And part of the reason we can't answer that question yet necessarily is because we don't have enough data, because so many of the studies have been done on polystyrene, what you get your coffee, you think about your classic styrofoam coffee cup, or polyethylene, these are plastic types that are really easy to buy from scientific companies to use in experiments, but we have a lot less information on kind of the real shapes and sizes and types of plastics that you find in a fish, or in my student's case in otter scat, for example. So we think that the shape can cause more toxicity. So like I was talking about, fibers can potentially be more toxic, or the smaller sizes that can more easily get trapped in tissues and are harder to excrete. Those can be more toxic potentially, maybe more likely to cause oxidative stress. But I would say we're still working towards that answer as to which are more toxic. But I think a lot of people think that bioplastics are potentially the answer. Oh, if we just make the plastic out of corn, or avocado pits, or whatever the latest trend is, that'll be better. But it turns out also that those plastic types are pretty persistent unless they're industrially composted. And so the answer is that they're all probably not great, but we're still trying to figure out which ones maybe are a little bit better, and maybe those are the ones we should make more products out of. Oh. Sorry, I don't have any one-word answers. I wish I did. No.
Great. Natalie, I do see your second question. I'm going to go to the questions for folks that didn't ask any first, and then I'll come back to that. So Dennis asks, are the wave actions and chemicals causing plastics particles to get smaller and less round?
Yeah, so plastics change in shape, they can change in size, they break down into smaller pieces the longer they're in the environment. They don't necessarily go away. And the thinking is that most plastic that's ever been produced is still out there somewhere, just a lot smaller maybe than it was when it was used. But yeah, they do break down via wave and wind action, biofilms that grow on them. There's all sorts of ways, UV light, that they break down into smaller pieces. Mm-hmm.
Great. Yvonne, who is our amazing social media guru for 500 Women Scientists, has a question. So Yvonne says, first, great talk. And for Sarah, sorry if you said this and we missed it, but do you also look at whether the fish spend more time near the edges of the wells versus in the middle of the wells, and looking at other different kinds of behavior besides maybe that freezing behavior you talked about?
Yeah, thanks, Yvonne. It's a good question. So we do look at a lot of different types of behavior. So we look at freezing, definitely whether they're on the edge of the well versus in the center, distance moved, velocity, and so a lot of different kinds of behavior as well. And something that I didn't get into too much, but is really interesting is that there are different types of pyrethroids that can cause different neurological effects. And I see a lot of differences in the changes in where they are in the well versus their velocity that they're moving, and the freezing as well, depending on what type of neurological change that pesticide is causing to the fish. So in short, yes, lots of different behavioral changes are looked at.
And adding to what Sarah said, we look at a similar diversity of behavioral changes in the fish exposed to micro and nanoplastics too. So, great question.
Great. So I'll go back to Natalie's question now, and I might summarize a little. So Natalie is asking, so she said she's been on personal vendetta against plastic bottles and unnecessary plastics for quite a while now, but she's been wondering about kind of the trade-offs and balance between some of these like vegan leathers and petroleum-based products that are meant to kind of take away impacts from other types of pollutant systems, and what that means for, you know, other balancing between these different types of pollutants. So again, her main question was about like, okay, how do we think about these other alternatives to be better for the environment that may actually have impacts from a plastics standpoint? Hopefully I summarized that okay, Natalie.
Yeah, and vegan leather, my understanding that vegan leather is essentially plastic. I can't say that I know a lot. Yeah, Sarah's nodding her head. So it's not great. I mean, it is taking the impact away from, you know, if you feel strongly about animal rights and not harming animals and using products made from animals, I can totally understand why you would want to choose to opt for something like vegan leather. But I think really maybe the bigger thing to think about is trying to reuse and to purchase things that are pre-loved rather than opting for new materials where you can. I think that's maybe taking a step back and thinking about, you know, shopping at thrift stores or shopping at ThredUp online to get your clothes, or, you know, your purses or whatever you're in the need of, instead of opting for one of those alternative materials is maybe the better option. But there is a lot of research going on right now, looking at how we can produce fabrics more sustainably and what the best materials are to be using. I saw a presentation a year and a half or so ago where a scientist was talking about producing fabric out of casein, which is a protein in dairy products. And so basically this was happening in Germany, I think, but they were taking milk from grocery stores that had spoiled, that had gone bad, and then they were extracting the casein from this milk and using that to make fabric, which is kind of mind-blowing. But we're trying to argue that maybe trying to capitalize on all this other waste we create and making products out of that somehow is maybe the way to go, rather than starting from scratch. So there is a lot of research in that area, and most of it I'm not 100% aware of, but that's one example.
Great, thanks. And I know we're approaching about 8:15, so we'll wrap up with one more question maybe, so folks can get on with their evenings, head on to the next thing. So a question from Kirsten, I think I'm pronouncing that correctly. Have you looked at mortality in the small critters, or only the behavioral changes? So in terms of some of these, you know, impacts that you look at, is it mostly behavioral or mortality as well?
So I can talk about that a little bit. So especially with salinity changes and thinking about those differences in the estuaries, we have looked at mortality, not with the same class of compounds that I was talking about tonight, but or I guess actually some of them are similar to what I was talking about tonight, but a really wide variety of compounds. I've looked at that, and we do see changes in mortality, at what concentration mortality occurs depending on the salinity. And sometimes that can be, you know, as high as a magnitude higher. So maybe you see mortality at 0.1 nanograms per liter, and then at a higher salinity, you see it at 1 nanogram per liter. And so that can be a pretty substantial difference depending on where you are in an estuary, if you're a fish or a small critter, like you said. So we do look at that as well.
And for plastics, it's a little bit different, and I can let Samreen speak to this too, but for the most part, you know, you're not seeing a lot of mortality at the concentrations of plastics that are found in the environment right now. And so we tend to aim on the lower end to be more environmentally relevant. So that's why we're looking at more subtle responses like changes in behavior, which can ultimately cause changes at larger scales. But Samreen, did you have anything to add to that on what you're
Yeah, it's
working
like
on?
same thing as you said, like mortality does not happen until you reach to a really high level, because there is a particle effect in addition to plastics itself. So my like little particles, they don't affect that much to the mortality. They do change the neurological problems that's cause behavioral changes, but not to the mortality until you reach really high levels.
Yeah. It's a great question.
Thank you. So since we are a little bit over time, I think we'll wrap up there. There's no more questions coming in. So thank you all again for your great presentation. Mary, is there anything else you want to say before we wrap up, or hopefully just see folks at our next TAP Talk?
Yes, we'll see you at the next TAP Talk. And thank you everybody for having a great talk, having 3 speakers. It's always very inspiring, especially when you get like the professor and the graduate fellow and the graduate student, actually postdoctoral fellow, I correct myself, seeing their careers and what they work on. I always learn so much from these talks, and it's always an inspiration to me. And we will see you hopefully next month, April, first Tuesday in April, whichever date that is, at 7 o'clock. Starting off at 6:45 with trivia. If you're around for that, we'd love to see you. And with that, I will bid you fair night. Have a great month.
Thank you all for having us. It's been a pleasure.
So I'm going to finish recording now, so you all know. I'm going to cut you all off. But thanks to everybody. Thanks, Vic, for being here. Susan, everyone. Take care. Bye, Susan.